Good morning children, welcome to today's session. I am Kalpana working as JL Zoology in COE GV. In today's session we are going to learn about types of immunity based on the types of responses evoked. They are humoral immunity and cell mediated immunity. Coming to humoral immunity, it is mediated by antibodies. Whenever antigen is entering into our body, B cells are stimulated so that the antibodies are released into the body fluids, and these body fluids are known as humors. And this type of immunity is known as humoral immunity. In secondary lymphoid organs, antigen binds with the FEB portion of the antibody which are present on the B cells, and these B cells engulf the antigen and destroy them and display the antigenic fragments with the help of the MHC2 class 2 molecules, and these are recognized by specific T helper cells, and they interact with the antigen and major histocompatibility class 2 complex, thereby releasing the interleukins, and these released interleukins stimulates the B cells so that B cells differentiate into plasma cells and memory cells, and the plasma cells are releasing specific antibodies specific to the antigen entered into our body, and these antibodies are released into the plasma and extracellular fluid, and by releasing the antibodies, antigens are immobilized and opsonized. Opsonizing means in which antigen structure is altered so that they become more susceptible to phagocytosis, thereby neutralizing the antigen which is entered into our body. Insoluble antigens are agglutinated, whereas soluble antigens are precipitated. In this process, there is activation of phagocytes as well as complement system. And coming to the second type of immunity, cell-mediated immunity. The name itself indicates it is mediated by cells, and the cells involved are T cells and the natural killer cells. And it is more effective, and it can detect the exogenous as well as endogenous antigens. And in this process, antibodies are not involved. The T cells can recognize the antigen which are presented by antigen presenting cells, and these antigen presenting cells specifically detect the exogenous antigen, whereas altered cells can detect the endogenous antigen. After the detection, these processed antigens which are processed by antigen presenting cells and altered cells these cells recognize these antigens recognized by the T cells, and the T cells are differentiated into activated helper T cells and memory T cells. These activated T cells or helper T cells secrete interleukins. These interleukins transform the activated cytotoxic T cells into cytotoxic T lymphocytes, or these are also known as killer cells, and these CTLs attack the infected cells, thereby releasing two enzymes, perforins and granzymes, and these perforins are making holes in the infected cells so that the granzymes are entering into the cells and activating certain types of proteins which are known as caspases, which are helpful in destruction of the cell through apoptosis. Apoptosis means it is a programmed cell death or cell suicide. In this, natural killer cells are acting in the same way as cytotoxic T lymphocytes, whereas these natural killer cells are acting in an antibody independent way, whereas CTLs are acting in antibody independent way. Children, try to find out the differences between the humoral immunity and cell mediated immunity. Thank you children.